G'day guys, just another one on MK4, Menaquinone-4. In this regard, it's related to the brain, and it's related also to LPS, lipopolysaccharides. I'll just share this for a sec. Yeah. So basically, Menaquinone-4 suppresses lipopolysaccharide induced inflammation in MG6 mouse microglia derived cells by inhibiting the nucleotide factor kappa B signaling pathway. So this is another Japanese study. Nothing like the Japanese to do good research. Let's just read the abstract. The overreaction of microglia is known to trigger inflammatory reactions in the central nervous system, which ultimately induces neuroinflammation disorders, including Alzheimer's disease. Now, that's sort of glia type cells. It's basically a macrophage. Think macrophage in the central nervous system, in the brain. I can't remember how, it's well over 10%. Of that, and it just sort of, it's involved in basically a lot of the regulation of the inflammatory system. That's why another reason why brain inflammation also requires vitamin D, because basically the macrophage, this type of microglia, also has vitamin D receptors. But today we're looking at basically not the regulatory side of things. We're looking at basically the role in controlling the overreaction of the immune system by Menaquinone-4. However, increasing evidence has shown that Menaquinone-4, a subtype of vitamin K2, can attenuate inflammation in the peripheral system. That includes other peripheral systems, but that's for another day. Whereas it was also observed at high levels within the brain, its function in this organ has not been well characterized. Well, the research is pretty piss poor out there, unfortunately, especially in North America and parts of Europe. I mean, if they're not doing something for a drug company, an actual fat soluble vitamin is much less important to them. Which is, therefore, we investigate the effects of MK4 on microglia activation, clarified the underlying mechanism. Mouse microglia derived MG6 cells were exposed to lipopolysaccharides, LPS, either with or without MK4 pretreatment. Cell response with inflammation to cytokines. So we'll just cover them. Interleukin-1B, tumor necrosis factor A, interleukin-6 were measured by QRT-PCR, which is a method. Bart knows quite about that method. I just know it in general, but it's probably for another day to go into the intricacies of the method. We further analysed the phosphorylation. I mean, you guys have heard of, phosphorylation. You understand it. So it has to do with the, biochemical processes which involve phosphates. Adenosine triphosphate. In the mitochondria, ATP. So similar phosphorylation of these enzymes. So we're looking at TAK1, IKKAE, and P65 of the NF-kappa B subunit of the Western blotting. We observed that in LPS-induced MG6 cells, the MK4 dose dependence suppressed the upregulation of these inflammatory cytokines. Yes. So let me just anyway, just wanted to make sure that that was the, yeah. Sometimes trying to remember some of these components, you know, that are sort of coming to transcription is always interesting, but that's not a good diagram. I think I'll give that a miss. Not finding the right sort of diagrams to basically make the point. But let's quickly, you can see. You can see some of these pathways that are involved. Okay. Now, because that's, I'm just going in a tangent, but you know, there are these subcomponents. I mean, if you want to go into them and look at all the biochemical pathways that they follow and all that, by all means, look into them. But all these things and how they get transcribed in their enzymatic components and all that isn't what I'm trying to focus on. Anyway, let's get back to looking at that further. The nuclear translocation of NF-kappa B in LPS induced MG6 cells was inhibited by MK4. So that's the big thing that, you know, the important thing here for the average person out there that doesn't need to understand the biochemistry. You know, what we're talking about is the inhibition of the overreaction of the immune system when LPS is around and circulating. And this is in particular with people that basically, you know, may have consumed a lot of crap in the past, may have leaky gut as a consequence of that crap, and now have some level of vulnerability to LPS. So LPS is going to be circulating, you know, until you basically heal your gut, you're going to get some LPS. Now, LPS is a purpose. Now, what's the purpose of LPS? Why do we have this lipopolysaccharides at the end? Like, it's a little component that basically is made up of, you know, sugar at the tip and, you know, sugar molecules that basically are coating these protein fat type structures that basically have an important role in the bacteria, in little organisms that basically want to, you know, create a certain electrical charge. And that's what they do. You know, like in your endothelial cells, we've got there is a negative charge that actually allows it to basically, you know, and the negative charge in the pericellular matrix that I've talked about in the past. Well, what LPS does for the bacteria, it actually allows the bacteria to actually have that sort of charge, so it doesn't stick, you know. Because if you're, just think of it, if you're a little cute bacteria, well, you don't want to stick. you want to be able to swim around, get around, do things and access things and whatever else. If you end up sticking with your mates, or, while they're going to eat some of your food, or you end up sticking where you shouldn't stick and stuff like that, it can be problematic for them. So LPS has this, a lot of people haven't discussed it, but that's what it is. Lipopolysaccharides, this little thing, is part of the pericellular matrix of the bacteria that allows the bacteria to apply a negative charge to its outer hairline structure. So that's basically what it is. But obviously, because it basically, it's like any organism, it's like your skin, like the outer coating of your skin or whatever, it does shed some of this stuff. Now, as long as you're shedding it in the actual intestinal tract and not in the blood, you don't have leaky gut, so your tight junctions are good because of your fat soluble vitamins that are basically maintaining those tight junctions of the mucosal layers at the gut barrier, and you're not getting, you haven't assaulted it with basically, lectins and basically, gluten and all these sort of things that a lot of us do or have done in the past, or still have the after effects of all that. What we need to do is basically deal with this problem, to basically reduce as we're healing our gut, we need to basically reduce the load of LPS in our bloodstream, because it's going to get past the blood-brain barrier, it's going to get into the brain and other tissue and cause inflammation. So this is where you require a high dose of MK4, Menaquinone-4, to basically deal with this inflammation until you fix your gut. And then you can come down, so that's what I'm trying to promote here. So I'm just giving you the information. Anyway, so that's covered. Now, I'm not going to read all this because you want to see. Okay, this is the actual dosages, various dosages of MK4, 0 to 50, what's it called, the micromoles, were administered within 24 hours. Okay, now let's just what was it, 25 was 13 to 26. I did the calculation earlier. It's basically, that's the amount of in micrograms. So a rat is about 30 grams. So it's 30 grams is about 0.03 kilograms. So let's say an average person who's 70 kilograms divided by 0.03, fuck it, 0.03. So it's about, a human is about 2,333.33 times an average person in size. So times in micrograms, which basically comes to nearly 61 micrograms, is the human equivalent dose in that regard. So you can get that very easily from animal foods, MK4 in that quantities. I've shown you before, we can very easily get those sort of quantities in that regard. So this is why a species appropriate diet, guys, is basically protective of the brain, protective of human tissue from high levels of inflammation due to LPS. So we don't need to worry as much about lipopolysaccharides like Gundry worries. Well, he should fucking start eating some meat if he's worried about LPS, because basically his species' inappropriate diet full of vegetables and shit basically doesn't really help him. Now, does it? It just malnourishes him and many of his other supporters. What we need is good animal products that are high in Menaquinone-4 that basically provide the protection if you even get a small amount of LPS. But because we're eating a species appropriate diet, we will get more than enough to basically mitigate these issues. So let's just look at a couple of these. People can go back and check themselves. Obviously, MK4 group, the control. So we've got interleukin-1B, tumor necrosis factor A, and interleukin-6. These are the big ones. And as you can see, pretty much 12 hours, interleukin-6 is quite suppressed. So it varies with different things. It takes a bit longer to suppress some of them at different levels. But as you can see, compared to the control, MK4 is doing a pretty good job. That's the important thing. And as you can see, all these which have been administered Menaquinone-4 to different levels, and you notice a dose response. This is important. So from 0, when we go to 0.1 to 1 and 10, you can actually see that the levels come down. So this fulfills the Bradford-Hill criteria of dose response. So these things will come down, and obviously if you remove the Menaquinone-4, they will go up accordingly over a certain length of time within the day. So that's what we want to see, dose response. The more Menaquinone, the more lower the levels, particularly when you get to these levels, like the higher levels. And tumor necrosis factor A, but interleukin-1B, that's quite a bit. Even at lower levels, it actually will suppress that quite a bit. So you don't need as high for that. But obviously, because there are going to be the other cytokines, it's best to get a better dose to cover them all. So this is just telling us that some respond much better. Some are suppressed much better by MK4 than others, So when we're looking at these ones as well, in terms of their differences, so between the different Ks, when it comes to this one, there's not a big difference. They can vary all over the place. So statistically not in any significant way. So when we look at control and we look at MK, those levels are much lower, including P65, which is another biggie in terms of, again here, MK4 control, more or less, LPS, much better improved when you've got both MK4 and LPS. Not as good as pure MK4 or the control, but but that's marginal, statistically insignificant. There is a difference there. When we look at P65 nuclear, you can see quite clearly control that's clean completely, hasn't been sort of affected. The one that it's more or less the same, not a difference. Again, it's come down. So basically, when you're dealing with lipopolysaccharides and these LPSs, or as Gundry calls them, little pieces of shit, on a species appropriate diet, you will get sufficient menaquinone to deal with them. It's another reason why we notice that people that have got brain fog when they're on a carnivore diet improve. It's another reason why we notice them when people get on a carnivore diet, their gut heals. their inflammation reduces. It's because basically MK4 that's coming in is playing a significant role in dealing with, first of all, the diet is less inflammatory. It doesn't have the inflammation that the other diets have. All eating causes some level of inflammation, okay? The moment you undertake oxidative phosphorylation at the mitochondrial level and you oxidize anything, you will create some reactive oxygen species. You will create some level of low-grade inflammation. That is a normal physiological thing that happens. There's no free lunch, but there's a fundamental difference between eating a species-appropriate diet that actually induces very low grade that can be eliminated more or less. This is why we get these extraordinary low levels. When somebody's healthy on a carnivore diet, they don't have any underlying conditions, and they're on a carnivore diet, their biomarkers are fantastic. We've seen it, many people that have shown their biomarkers, absolutely fantastic, because even the small amount of inflammation just from general living can be severely suppressed by, when you eat a species appropriate diet, you get the fat soluble vitamins in there, which are critical. And that's what we notice. And that's the important take home message here, guys, that it's, forget about all the mumbo jumbo biochemistry and all that we can talk about this reductionist pathway and that one and whatever. But the take home message is here that eating animal foods is so protective to the body. It produces very low grades level of reactive oxygen species, very little inflammation, and the nutrient density of the components that you take in have a strong suppressive effect against these inflammatory pathways. And they regulate them well, like in particular, when we're looking at, the fat soluble vitamins, vitamin D, vitamin MK4 and retinol that play a big role in this sort of regulatory sort of side of things. So that's sort of the take-home message. I hope you enjoyed this about LPS, lipopolysaccharides. So the next time a vegetard out there tells you, makes some issue, or somebody from the Gundry sort of mafia group, plant-based mafia group, try to tell you, that how important it is to eat their type of foods that will protect you, you just tell them, I get plenty of MK4, mate, and I can basically keep all those inflammatory markers under control in that regard. And, I've got the taurine in there that basically is clearing out the beta amyloid, that basically I've got the MK4 that's keeping all that sort of under control so it doesn't get out of hand, and keeping the inflammation down. So you tell them that I don't need to do some species inappropriate diet or take some special supplements or whatever, or you know what I'm talking about. Basically, I just eat meat and I'm fucking healthy. Anyway, hope you enjoyed it. See yous.